So we continue talking about bone tissue in chapter 6. In the last recording, we talked about compact versus spongy bone, and then we talked about bone development. We just introduced intramembranous ossification as a type of bone development, and then we went into more details about endochondral ossification, which is how long bones are formed. Again, we're going to save intramembranous ossification for the next module when we talk about flat bones of the skull, even though we do talk about the sternum and ribs in this module. Um, so after, if you recall, ossification involves development, it involves growth, and then in childhood and then in adulthood, it's primarily involves remodeling or repair. So if we go back, we're going to talk about growth now, which is what happens during childhood, soon from birth to adolescence. And this fifth stage is where we sort of touched on that with bone development, where there is an epiphyseal plate located at the proximal and distal metaphyses, composed of hyaline cartilage, and that persists throughout childhood as the source of bone elongation here. And then into adulthood, once the growth has completed, the ossification, which is actually proceeding sort of from this area up through the cartilage or from this area down through this cartilage, once it reaches that growing front, it will completely ossify the whole plate and become this epiphyseal line in adulthood once the growth is finished. All right, so but when we're looking at that epiphyseal plate, we're looking at the chondrocytes here in the interior, just as we've talked about before, that divide and hypertrophy and die, and then the resulting ossification, line of ossification will come in and invade that area. So that's kind of what we're going to focus on here. So that is what we just described there is called interstitial growth, which we'll describe again a little bit here, which is growth in the length of the bone. So that's one type of bone growth we could talk about. And then after we talk about interstitial growth, we'll talk about appositional growth of bone, which is growth in the diameter, like laying down additional layers of a tree trunk, essentially. And appositional growth continues through childhood and then a little bit into adulthood as well. All right, so let's talk about interstitial growth. And these are the same terms we used with the growth of cartilage, if you recall. So growth from the interior versus growth from the exterior is kind of how those two terms apply. So with interstitial growth of long bones, we have that layer of hyaline cartilage at the in the metaphysis that we just looked at in that diagram back there, which is the site of bone lengthening, which we'll talk about how that happens here in 5 distinct microscopic zones. So here's our 5 distinct microscopic zones of the epiphyseal plate, and I might give you some context here. The proximal epiphysis, if we're talking about, say, growth of the humerus, the proximal epiphysis of the humerus would be up in this area up here, whereas the diaphysis of the humerus would be down here. So we're going to be watching growth occur sort of up through the slide. And so that's demonstrated in this proximal epiphysis here. So we'll start with this, actually up here with the zone of resting cartilage, which would be kind of up towards the proximal epiphysis. And the zone of the zone of resting cartilage is the furthest away from the medullary cavity of the diaphysis. And it consists of the small chondrocytes here, small chondrocytes within a matrix of hyaline cartilage, within that their extracellular matrix. Now the zone of proliferating cartilage means the zone of dividing cells. Here the chondrocytes are slightly larger and they're undergoing rapid mitotic division. So you can see they start to line up like stacks of coins here because they're dividing in sort of an upward direction. And so then the zone of hypertrophic cartilage is shown here, where now the chondrocytes have stopped dividing and they become enlarged or hypertrophied. which is a sign that they're soon going to die here. The zone of calcified cartilage, you can see now where the matrix has calcified, so the chondrocytes have died. They don't have that source of nutrients to help support them, so they basically are disappearing from the lacunae. So we have lots of empty lacunae here. And so what happens is that minerals get deposited between the stacks of lacunae, which is what kills the chondrocytes. And then the zone of ossification now is where the walls between the lacunae have broken down, forming these longitudinal channels. And then those channels become invaded with capillaries, which are bringing osteoprogenitor cells to this zone. And those osteoprogenitor cells, like we've talked about previously, are going to differentiate into osteoblasts, which then will start to produce osteoid on top of that calcified cartilage. So the newest cells are up here at the top, having just been formed from mitosis, and then the older cells or chondrocytes are kind of here at the bottom. So this is sort of a snapshot in a whole dynamic of a moving front of osteocytes or osteoblasts kind of invading spaces. So you've got to think of these mitotically active chondrocytes kind of adding proximally to the epiphyseal plate, while the osteoblasts are here distal to the epiphyseal plate, trying to catch up with that proliferating front. And so that's going to cause the bone to elongate in the upward or peripheral direction, or sorry, the proximal direction. Now there is a condition that is referred to as achondroplasia, and I don't have it listed here in the slide, where it's a genetic disorder, whereas the second and third zone are inactive in the limbs of individuals with this disorder, resulting in shorter limbs, but the body parts are typically sized everywhere else. So individuals with achondroplasia may be referred to as little people or other things that is caused by a spontaneous mutation that is inherited from an autosomal dominant gene. Now this shows another example of an anomaly related to bone lengthening, which was the Giant of Illinois in between 1918 and 1940. Robert Pershing Wadlow was basically considered the tallest man at that time. 8'11" was his height. His father, by comparison, here on the left is 5'11", sort of a more typical height for our generations. And so giantism was his condition, and is caused by an enlarged pituitary gland, which is producing an overabundance of growth hormone. Now, growth hormone stimulates the cartilage production in that zone of proliferation in the epiphyses, in the epiphyseal plates rather. He had to wear leg braces to help support his weight, as in this height here, and he died early at age 22 from infections caused by blistering from those braces. All right. The shortest person in the world was Ping Ping, who's no longer living, at 29 inches tall, and he lived to about age 20. So you can see by comparison to the woman here, he's about knee height. And his condition is osteogenesis imperfecta, and there are 7 types. This is referred to as brittle bone disease. 7 types of this sort of disorder. It's a genetic disorder where certain types of collagen, different kinds of collagen, cannot be produced. So the different types depend on the quality or the quantity of the collagen that can be produced. And again, it's an autosomal dominant disorder. So those are just examples to apply to our bone elongation. Now here's apositional growth. So we just finished interstitial growth. We're moving into apositional growth. And apositional growth depends on the osteoblast versus the osteoclast activity. So apositional growth is growth in the diameter of long bones. So you can see over time that the diameter is getting larger. It occurs, it's based on the functioning of the osteoblasts within the periosteum that produce bone matrix, increasing the circumference of the bone, and therefore the diameter. And sort of, you can see here between infancy and childhood, the osteoblast activity would be quite active here. As is the osteoclast activity, or basically just beginning. The osteoclast activity is going to hollow out that medullary cavity so that it keeps up with the growing circumference or the diameter of the long bone. And so then into the young adulthood, you can see the medullary cavity getting even larger. So bone resorption is happening in the same direction as bone formation. Osteoclasts resorbing bone, osteoblasts producing bone. And so this tends to then be kind of similar to what we end up with as a full mature adult. So in childhood, you can sort of rate the level of activity of osteoblasts versus osteoclasts. In childhood, the osteoblast activity is greater than the osteoclast activity. Then in young adulthood, they're about equal. And then in older adulthood, you can see the osteoclast activity it's caught up here with the compact bone. So the osteoclast activity is now outpacing the osteoblast activity. So again, this is hormonally controlled through life, which we'll talk about here in a couple of slides. Now, similar to bone remodeling, injuries and other mechanical stresses can result in bone remodeling. So the bone is constantly being remodeled due to stress on bones, whether we're talking about injuries or the degree of exercise someone may have. Where osteoblasts lay down bone matrix and osteoclasts are resorbing bone matrix. And remember, the osteoclasts, by resorbing bone, are releasing calcium and phosphorus back into the blood. And so that may be important for just maintaining blood levels of calcium and phosphorus so that those can be supplied elsewhere in the body for things like muscle contraction. Calcium is important for muscle contraction and nerve impulse conduction. And as we said earlier, phosphorus is important for ATP and DNA synthesis. The mechanical stresses associated with muscle contractions as well as gravitational forces increase bone density by increasing osteoblast activity. And when osteoblast activity is sufficient, that means the collagen production is continuing. And as a result, hormones will increase the calcium and phosphate deposition. So the more osteoblast activity there is, the more collagen production there is. And as you recall, calcification, laying down the calcium and phosphorus salts or depositing them, relies on the presence of those collagen fibers to sort of crystallize on the surface of. Now, athletes have greater bone density because of the mechanical stresses involved in the exercise. So that's why women and older adults in general are encouraged to exercise. A lack of exercise causes the demineralization of bone and reduces collagen formation, which you can see when someone gets a cast taken off for a broken bone, and astronauts in space are at risk of losing bone density. And it also increases the risk of osteoporosis, which we'll talk briefly about. Now there's an extensive arterial supply through the periosteum and the nutrient foramen, which are the nutrient arteries here, which bring nutrients and oxygen to bone and take away waste. They also circulate hormones that are needed to signal bone growth and repair. And certainly nerves in bone will also signal the need for injury and repair to bones. Bone and aging, there are two different things that are happening with bone and aging. First of all, bone loses its ability to produce organic matrix, which is the result of the declining osteoblast activity, while osteoclast activity maintains its normal level of activity. And then bone also loses calcium and other bone salts. So bones become weaker and there's less collagen present for deposition of calcium and phosphorus. And this can result in osteoporosis, which is the loss of bone density, which you can see here on the right compared to on the left. So this is primarily what we'll be covering for this chapter and concludes the recorded lectures.